Hey guys, what's up? My name's Thomas. I'm going to hopefully provide a video for you that is the only carbohydrate metabolism video you will ever need. So in this video, we're going to talk about GLUT4, GLUT2. We're going to talk about glycolysis, gluconeogenesis, glycogenolysis, fructose, galactose, gluconeogenesis, citric acid cycle, electron transport chain, pentose phosphate pathway, and oxidative phosphorylation. That should cover everything you should possibly need for carbohydrate metabolism on the MCAT. As you guys know, this is a very important topic, and hopefully I'm going to be able to provide this for you in a way that includes all the relevant information you need without all of the fluff that some of the big name third-party brands will include. Now, obviously, I learned from those brands myself, and I've kind of learned that if you can teach something well, you really mastered the subject. So my goal here is that I will provide you all of the information I use to attain a very high percentile score on the MCAT by kind of going through my process of figuring out what information was actually needed and what wasn't. So without further ado, we're going to begin. So I would like to introduce that first and foremost. Carbohydrate metabolism is the breaking or building of carbohydrates. And so this can really be categorized into two different topics. We have the catabolic right here, and we have the anabolic right here. Now, to really make this simple for you, think of catabolic processes as a breakdown. We are breaking down a complex molecule into simple molecules, and a prime example is glycolysis. We are taking a molecule of glucose and we are breaking it down into pyruvate. So we're going from a 6-carbon molecule to 2 3-carbon molecules. That is catabolic. A way I like to remember this is by thinking of a cat with this beautiful drawing of a cat I made. Cats tend to be destructive. They claw at things. They're going to break things apart. So a catabolic reaction is something that breaks something down. Likewise, we are on the anabolic end here now. Anabolic processes means we like to build a complex molecule from simple molecules. And an example is gluconeogenesis. Now, in gluconeogenesis, we are doing reverse glycolysis, but not really. We'll cover that later, but we're taking pyruvate molecules, which are more simple, and we're creating glucose, which is a more complex 6-carbon sugar. We are building a complex molecule from simple molecules. So anyway, that's just our brief overview on what carbohydrate metabolism really encapsulates. It's not a black or white thing where it's always catabolic, it's always anabolic. There's a very complex interplay of these processes that depend on cellular conditions of energy and our fed versus fasted state, and we're going to cover all of that. So anyway, we should first begin by asking, how does glucose even enter our body cells? I think it's a very important thing to know that students often overlook. Well, the way we do this is we have these transporters known as GLUT transporters, and we have GLUT 1, 2, 3, and 4, although GLUT 1 and 3 aren't really that cool, so we don't really tend to focus on them. Our main focus is GLUT 2 and GLUT 4, and I have conveniently categorized some things that we can compare GLUT 2 and 4, such as the location, the Km, the Michaelis-Menten constant, and the kinetics. Now, I'm going to go over this. You guys, I also would like to preface that you could screenshot these notes and take more of what I'm saying. These are very generic things that just serve as like a pacing thing for me to explain and talk over. But with that out of the way, GLUT 2 is located in the liver and the pancreas, specifically the beta islet cells of the pancreas. They tend to have a high Michaelis-Menten constant, and if you recall from enzyme kinetics, a high Michaelis-Menten constant means that we have a low affinity for glucose. The kinetics follow first-order kinetics, which means they are proportional to glucose concentration. You don't need to worry about what this means right now, because once I talk about GLUT4, I'm going to give you some comparative points to look at. As for GLUT4, the location is in skeletal muscles and adipocytes, which are just fat cells, and the Km is relatively low, which means we actually have a high affinity for glucose. In terms of kinetics, GLUT4 follows zero-order kinetics, which means it's not proportional to glucose concentration. Now, the big thing I'd like to focus is on kinetics right here, right? So this kind of dictates function for liver cells, pancreas cells, skeletal muscle cells, and adipocytes. I want you to think of what this Km and this Km here mean. So a high Km in GLUT2 means that we have a very low affinity for glucose, but we follow first order kinetics, which means we have a intake of glucose that is proportional to our glucose concentration in the blood. And what this means is that when our blood glucose concentrations are at a relatively physiological normal state, our liver and our pancreas isn't really going to be taking in much of that, so that the glucose can go to other important things like our muscles for movement and our cardiomyocytes for, heart contraction and our adipocytes for fat stores. That is why the Km of GLUT4 is relatively low, which means it will tend to bind glucose at a lower concentration in the blood than GLUT4 will. But it follows zero order kinetics, which means it's not proportional to glucose concentration like GLUT2 is. What this means is that no matter what number of molecules of glucose are in the blood, that transporter will take in a set amount, regardless, as long as the affinity is at or above the Km. And so what this means is that the only way to take in more glucose with the GLUT4 transporter is to express more GLUT4, and that would require some transcriptional activation of the mRNA transcript. That's some DNA and RNA biotech. I will maybe make a video on that. It's another high yield topic. But know that the only way for GLUT4 to take in more glucose is to physically express and traffic more of those GLUT4 transporters to the cell membrane, whereas GLUT2 is proportional, meaning the more glucose is in the blood, the more glucose the liver and pancreas will take in. And you'll learn why the liver is very special in carbohydrate metabolism. But anyway, now that glucose has entered the cell and we've learned how it enters the cell, what do? Well, the short answer is that a lot can happen. And what I've illustrated here is a very, 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 keyword very generic pathway of what processes might happen for humans who are breathing air, which is all of us, right? We would undergo glycolysis, possibly. We would create acetyl-CoA, which can form from many roots. That acetyl-CoA could enter the citric acid cycle. The citric acid cycle would produce intermediates that can kind of push the electron transport chain forward. And the electron transport chain can, facilitate oxidative phosphorylation, which leads to tons of ATP. Now, it's really not that simple, keyword yet, because hopefully by the end of this video, it will be simple. But for now, it's not that simple. And so what I would like to start us off with is glycolysis. This is where most of the big name brands will introduce glycolysis, and I personally think it's the right place to introduce it as well. But what I've highlighted here in this big red box is that we are turning one glucose molecule into two pyruvate molecules. This is very important. What I've also highlighted here is this yellow line that I like to consider the dividing line between what I call the energy investment phase and the energy profit phase. And so this is a very generic breakdown that I've created that we can follow for how we get from glucose to pyruvate. And I will, in the next couple pages, talk about the regulation of key enzymes, the rate limiting enzyme, all this stuff. But for now, I would like to just talk you through how we get from glucose to two pyruvate molecules. So here we are with glucose. We've entered through a glucose transporter. And what we're going to do first to trap glucose in the cell is phosphorylate it. And we're going to phosphorylate this glucose into glucose 6-phosphate. And the enzyme that we're going to use to catalyze this reaction is hexokinase. Now, hexokinase will take an ATP molecule, and as a result of this phosphorylation, it will turn into an ADP molecule. This is kind of, if you're kind of getting the foreshadowing here, this is why I call it the energy investment phase, because we are using energy actually to push through glycolysis. And we know glycolysis does produce energy, so we do need to profit from it, and we will learn about that in the profit phase. But anyway, the next step is that we're going to isomerize glucose 6-phosphate into fructose 6-phosphate via the enzyme isomerase. Now, in my opinion, this is the most boring step of glycolysis. Nothing really matters here except for the fact that we're turning glucose 6-phosphate into fructose 6-phosphate. The same cannot be said, though, for the next step, which is going from fructose 6-phosphate to fructose 1,6-bisphosphate. And I've abbreviated that as fructose 1,6-BP. And the enzyme that catalyzes this is PFK1, which stands for phosphofructokinase 1. PFK1 will take a phosphate group from ATP and move it to fructose 1,6 bisphosphate, and as the result, we'll have an ADP molecule created. Fructose 1,6 bisphosphate is still a 6-carbon molecule, so what we need to do is cleave it into 2 3-carbon molecules, and this is done by the enzyme aldolase. Aldolase will create 2 3-carbon molecules, one of them of which is glyceraldehyde 3-phosphate, and the other of which is abbreviated as DAP, which stands for dihydroxyacetone phosphate. DAP is important in its own regard when it comes to serving as a feed point for other pathways to enter, as well as serving as a point for DAP to differentiate out of glycolysis into creating glycerol, which is a big aspect of the backbone for triglycerols. For now, though, we're going to focus on glyceraldehyde 3-phosphate because this is the molecule that pushes glycolysis forward. Do know that the concentrations of DAP and glyceraldehyde 3-phosphate are in an equilibrium in the cell, and that the enzyme that is interconverting these molecules is known as triose isomerase. That said, we're focusing on glyceraldehyde 3-phosphate, which I will be calling G3P sometimes. And so the next step is to turn glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. And this is abbreviated as 1,3-BPG. The enzyme that catalyzes this reaction is G3PDH, which is glyceraldehyde 3-phosphate dehydrogenase. Now, a light bulb should be going off in your head when you see the DH, because a dehydrogenase enzyme, for all of the enzyme experts out there, is typically going to be catalyzing an oxidation reduction reaction. And so in this case, our coenzyme NAD+ is going to be serving as the species that will be reduced into NADH. And so NADH is the first product that I like to be considered a profitable product. And we'll learn about NADH and its role as an electron carrier later, but do know that this is a very valuable product from glycolysis. In addition to the production of NADH, we are adding another phosphate group via the addition of an inorganic phosphate, which is PI, to this G3P to create 1,3-bisphosphoglycerate, 1,3-BPG. From here, 1,3-BPG is going to turn into 3-phosphoglycerate through the enzyme phosphoglycerate kinase. And again, light bulb should be going off in your head when you hear the word kinase. We are transferring a phosphate group to something, and that something is ADP. When we transfer a phosphate group to ADP, we get ATP. And this creates the first of 2 ATP molecules in this cycle. And this type of phosphorylation is known as a substrate-level phosphorylation. And it has a fancy name, but know that all that means is that it's an oxygen-independent phosphorylation, whereas the rest of the ATP you'll see produced in carbohydrate metabolism is dependent on oxygen, which is why it's called oxidative phosphorylation. This, however, is called substrate-level phosphorylation, and it's the first of 2 in glycolysis. That said, 3-phosphoglycerate will turn into 2-phosphoglycerate via the action of a mutase enzyme. This, next to the isomerase step, is another boring step where not much really happens. However, 2-phosphoglycerate will turn into a molecule known as phosphoenol pyruvate, known as PEP, and the enzyme that catalyzes this is enolase. Enolase is going to result in the production of a water molecule, and so phosphoenol pyruvate is now one step away from becoming pyruvate, and it will do so through the enzyme pyruvate kinase. Pyruvate kinase, as you can see here, will perform the second and final substrate-level phosphorylation to produce pyruvate. Now, I could end it here in glycolysis, but I did want to illustrate that pyruvate does have a couple of fates here that we'll talk about more, but some of which are lactate, acetyl-CoA production, and ethanol if you're like a yeast cell, which we're not. But anyway, this is the generic map of glycolysis. That said, I would like to talk about the 3 key regulators, which are hexokinase, phosphofructokinase 1, and pyruvate kinase. Now, with the first reaction, we have glucose going to glucose 6-phosphate through the mechanism of hexokinase, and this serves to trap glucose in the cell, like I've said. But how are we regulating hexokinase from working too fast or working too slow or not working at all? Well, the way we do this is by a form of feedback inhibition. And what feedback inhibition means is that the product of the enzyme or the product of a pathway further down is going to build up, and as it accumulates, that concentration will reach a concentration such that it will have an affinity that can bind to an enzyme and allosterically inhibit it or activate it. In this case, a high glucose 6-phosphate concentration will inhibit hexokinase and shut it down. It's basically saying, hey, we have enough glucose 6-phosphate already. We don't need any more. Just shut down. And that's what the hexokinase regulator is. The next and most important of glycolysis is the fructose 6-phosphate to fructose 1,6 bisphosphate step. And so this, I forgot to write this, so I'll write it in right now. Obviously, we have PFK1 performing this reaction. And so PFK1 is the rate-limiting enzyme of glycolysis. This means that this enzyme and this step determines the whole speed at which glycolysis can perform. And so there's a couple key regulators to PFK1. An increase or buildup of ATP or a buildup of citrate lead to the shutting down or inhibiting of phosphofructokinase 1. And so some of you guys can probably piece together why this is, and some might not be able to. But what I would like to say is that if we're high in ATP, that means that we, as a cell, are high in energy. And it means we don't really need to be producing any more energy because we know glycolysis produces pyruvate that will feed into the citric acid cycle, which will feed into the electron transport chain, which will feed into oxidative phosphorylation. And that results in even more ATP. So if we have high ATP already, we don't really need to do this. So we would shut down PFK1, thus shutting down glycolysis. The same can be said for citrate, which is a citric acid cycle intermediate. If you have a buildup of a citric acid cycle intermediate, it tends to mean that we are going to be trending in the path of producing tons of electron carriers that will fund the production of ATP. And so this also leads to the shutting down of PFK1 because it kind of tells the cell like, hey cell, we already have a lot of energy or we will have a lot of energy soon. No need to work on making more. In the case that we want to activate PFK1, we would need a high concentration of AMP or a high concentration of fructose 2,6 bisphosphate, which is kind of liver specific, but the big focus here is a buildup of AMP. A buildup of AMP indicates that the cell is energetically malnourished. It needs more energy because we want that AMP and that ADP to be ATP. So a buildup of AMP tells PFK, hey, we got to speed things up. We need to make more ATP. We need to start trending into the citric acid cycle, start working. And that is exactly what it does. It activates PFK1. I would like to focus on fructose 2,6 bisphosphate as an activator of PFK1 right over here in this yellow, which I've kind of illustrated as the liver. Now, fructose 6-phosphate actually has two fates. In this picture, I did kind of illustrate that fructose 6-phosphate goes directly to 1,6 bisphosphate, and the majority of it does. However, in the liver, we can go from fructose 6-phosphate to a molecule known as fructose 2,6 bisphosphate through PFK2, phosphofructokinase 2. What fructose 2,6 bisphosphate does is it can turn, as I wrote here, it can really turn on PFK1. And what I've also written here is that regardless of the energy state, it overrides PFK1. And so I want you to think about the liver, right? Like I've said, the liver displays first order kinetics, which means we'll be taking in tons of glucose when blood glucose is high. And so maybe taking in tons of glucose leads to a buildup of ATP. Normally the cell would shut down. But in the liver, we do things like glycogenolysis, we produce glycogen, we do all of these storage and buildup processes that we kind of want glycolysis to continue. So what we do is glucose in the liver will now be invested towards a shift in focus of storage and processing, such as glycogen, glycogenesis. And this way, the activation and overriding of PFK1 despite glucose levels and ATP levels in the cell can allow us to make acetyl-CoA for fatty acid synthesis, for example, which would be an energy storage process. And so this highlights, again, that PFK2 creates fructose 2,6 bisphosphate as a method to override the inhibitory actions of PFK1 despite energy level. That's typically only in the liver. It's a little bit of a mouthful, so if you need to watch it again or take more notes on this slide, that's perfectly fine. Anyway, the third and final regulatory enzyme in glycolysis is phosphoenolpyruvate to pyruvate by the enzyme pyruvate kinase. Now, this one has a very rare form of feedback known as feed-forward feedback. And this is when an earlier product of a pathway tends to stimulate and cause like anticipatory production and activity of an enzyme further down the pathway. And in this case, the product fructose 1,6-bisphosphate tells pyruvate kinase to kind of be prepared and be ready to metabolize because we're going to be building a product. In a very logical sense, the buildup of ATP indicates that we're a very energetically satisfied cell and that we don't need to undergo these energy producing processes. So that kind of tells us for pyruvate kinase to slow down. On to the next thing, I would like to briefly talk about glycolysis in red blood cells, because glycolysis in red blood cells is the only form of energy production that can occur. And that is because red blood cells don't have mitochondria. They're kind of losers, but they also keep us alive, so I guess they get a pass. But anyway, in glycolysis of red blood cells specifically, in the stage where we're at 1,3-BPG, we have a special enzyme known as phosphoglycerate mutase. Phosphoglycerate mutase results in the production of a molecule known as 2,3-bisphosphoglycerate. And so what 2,3-bisphosphoglycerate does is it binds to hemoglobin A, which is the oxygen-carrying protein of our red blood cells, and it causes a right shift in the oxyhemoglobin curve. I'm not going to draw the curve, but I will maybe go over this if I'm going over biology later in future videos. What this means is that the rightward shift enables hemoglobin A to be fully saturated in the lungs and allows dissociation in oxygen-depleted tissue. This basically allows the loading and unloading of oxygen in areas where it needs to be loaded and areas where it needs to be unloaded. So that's an important thing about glycolysis in red blood cells. I would like to emphasize, as I went in the drawing of glycolysis as a map, pyruvate does have options from glycolysis, and what I would like you to know is 3 options. Option 1 is the most common and the most tested, of pyruvate to acetyl-CoA. And pyruvate turns into acetyl-CoA through the pyruvate dehydrogenase complex, and there's a couple additional products and reactants that are happening here. An NAD plus molecule is required, and it will be reduced into an NADH, and we're also going to have a decarboxylation occur. And this makes sense because acetyl-CoA is a 2-carbon molecule, pyruvate is a 3-carbon molecule. Clearly, we had to lose a carbon, and so that's what the decarboxylation is. I would like to highlight that the pyruvate dehydrogenase complex is really 5 enzymes, 3 of which make acetyl-CoA. To be honest, the specifics of the 3 enzymes is not too important, but you do need to know that there are some cofactors and coenzymes that are required that I did not write here because it slipped my mind. And those 5 famous cofactors or coenzymes are as follows. So we need thiamine, we need FAD, we need lipoic acid, we need NAD+, and we need coenzyme A. And so those are the 5 cofactors or enzymes that the pyruvate dehydrogenase complex needs. The 2 other enzymes of the 5 are a kinase and a phosphatase. And what this means is they will either phosphorylate or dephosphorylate pyruvate dehydrogenase as a complex. The kinase will inhibit pyruvate dehydrogenase when ATP levels are high. And this kind of makes sense. If ATP levels are high, we don't need to be making acetyl-CoA that will enter the citric acid cycle and make even more ATP. So when ATP is high, we're going to shut it down. Likewise, the phosphatase will activate instead of inhibit pyruvate dehydrogenase when ADP is high. And this makes perfect sense because when ADP is high, we have tons of molecules that are just begging to be phosphorylated into ATP. And so pyruvate dehydrogenase will really want to speed up the production of acetyl-CoA so it can enter the citric acid cycle, so it can enter the electron transport chain and then produce tons of ATP. Likewise, we have another demonstration of feedback inhibition where buildup of acetyl-CoA tells us we're in an energetically satisfied state and thus we don't need any more. And that would inhibit pyruvate dehydrogenase. So these points of inhibition and activation are very important in understanding the complex. I think that's more important so than understanding what the 5 enzymes and their names are. The next step, or the next option of pyruvate, is to turn into lactate. And this is only in the case of anaerobic conditions. I want you to imagine a scenario where the cell is a finite amount of NAD plus and NADH that is existing in equilibrium. At some states, it's going to be high in NAD plus. At other states, it's going to be high in NADH. We call from glycolysis that one of the steps results in NAD plus turning into NADH through the mechanism of G3P dehydrogenase. Theoretically speaking, If we kept glycolysis going in anaerobic conditions, it would only continue until we run out of available NAD+. In other words, when all NAD+ has been converted to NADH, glycolysis would slow down or actually stop entirely, and this would lead to cell death in anaerobic conditions such as strenuous exercise, all that stuff. And we don't die when we exercise, so how do we combat this? Well, we produce a molecule called lactate. And to produce lactate, we need an enzyme known as lactate dehydrogenase. And lactate dehydrogenase solves that whole theoretical issue. It turns the NADH we produced from glycolysis back into NAD+ through an oxidation reduction reaction, as all dehydrogenases do. So in the case of NADH, it's being oxidized back to NAD+, and in the case of pyruvate, pyruvate is reduced into lactate. And lactate will build up as a waste product, and that's why sometimes the burning when you're doing tons of cardio is from lactic acid. And so that is in anaerobic conditions. The next option for pyruvate, option 3, is mostly in yeast cells, and we're not yeast cells, but it is good to know still, is that pyruvate can turn into acetaldehyde, which can turn into ethanol. And that is actually the basis of how some beers are made with yeast cells. But anyway, I would like to total and tally the ATP that we could in theory make through just glycolysis. So we know that 2 ATP are spent, we know that 4 are made, and we know that 2 NADH are made. This leads to a total of 2 net ATP, or 2 NADH. There's going to be a grand total calculation with aerobic metabolism included at the end, but for now, keep these 2 numbers in the back of your head. Now we can go on to gluconeogenesis, which is reverse glycolysis, kinda, not really, but that, for some reason books say it's reverse glycolysis because most of the steps are, but the 3 steps that aren't are quite different, and so we'll go over them all. In the same way that I illustrated glycolysis, I just would like to go over a quick map of gluconeogenesis, which is the conversion of pyruvate to glucose. Rather than starting at the top like I did in glycolysis, we'll be starting at the bottom. And the steps I've highlighted in yellow very boldly are the differentiating steps in glycolysis or in gluconeogenesis. So anyway, we have pyruvate in this mitochondria here. Pyruvate itself will turn into oxaloacetate through an enzyme known as pyruvate carboxylase. As the name implies, carboxylase will be adding carbon to a molecule. Pyruvate is a 3-carbon molecule, oxaloacetate is a 4-carbon molecule. To get that 4th carbon, we use pyruvate carboxylase. Oxaloacetate runs into an issue though, because it can't freely cross the mitochondrial membrane. To combat this, it'll quickly turn into malate, and this malate will exit the mitochondria through the malate aspartate shuttle, which I won't really be detailing too much, but it's a shuttle that allows malate to exit the cell. And so once malate exits the cell, it can reform into oxaloacetate. So this is where we're starting in gluconeogenesis. Oxaloacetate has exited the mitochondria, and it is now in the cytosol, the cytoplasm, and is ready to work its way back into a glucose molecule. What will happen is oxaloacetate will turn back into phosphoenolpyruvate, so kind of reverse glycolysis, with the enzyme PEPCK. This stands for phosphoenolpyruvate carboxykinase. It's a mouthful. Carboxykinase comes from the fact that we are using a GTP molecule, and as a result, we get a GDP molecule. And so phosphoenolpyruvate just undergoes reverse glycolysis here into 2-phosphoglycerate. Instead of producing a water molecule, we are inputting a water molecule. 2-phosphoglycerate turns into 3-phosphoglycerate by the same mutase enzyme. 3-phosphoglycerate turns into 1,3-bisphosphoglycerate through the same phosphoglycerate kinase enzyme. And the same is all the way, because I'm not going to repeat the steps of glycolysis in reverse, the same is all the way until we get to here. Once aldolase has turned G3P back into fructose 1,6-bisphosphate, to turn fructose 1,6-bisphosphate into fructose 6-phosphate, we use an enzyme called fructose 1,6-bisphosphatase instead of PFK1. This enzyme will result in the release of an inorganic phosphate. And that makes perfect sense because we went from 2 phosphates to a monophosphate. And then we will, reisomerize fructose 6-phosphate back into glucose 6-phosphate. Same boring step of glycolysis. But now is the final differentiating step. glucose 6-phosphate has to turn into glucose, and instead of hexokinase doing this, we're using glucose 6-phosphatase, and in the same way we release an inorganic phosphate, we are doing so here again. And so this is gluconeogenesis, very broadly speaking, but I would like to go into some key notes here. Pyruvate for gluconeogenesis is not the same pyruvate from glycolysis. Really ingrain that in your head. And the reason this is because pyruvate to acetyl-CoA through the pyruvate dehydrogenase complex is irreversible. What this means is the delta G is so negative for the forward reaction, which means spontaneous and energetically favorable, that it makes absolutely zero sense for the cell to even try to reverse this reaction. So it won't. So pyruvate to acetyl-CoA is not the same pyruvate that we got from glycolysis. Pyruvate can come from varying sources in the cell, such as the Cori cycle or the alanine cycle, much of which are kind of in a different, more like ketogenic, glucogenic amino acid metabolism, and I could go over that in another video, but know that pyruvate comes from varying sources for gluconeogenesis. The second point I would like to emphasize is this is not a perfect reversal of glycolysis, and this is due to the irreversible steps. I would like to further emphasize what an irreversible step is. Delta G in an irreversible step is so negative that it's highly, highly favorable to go in one direction, which is the forward direction. So much so that the reverse theoretically or essentially cannot happen. So gluconeogenesis uses different enzymes to circumvent this. Circumvent just means to overcome this. So to overcome the delta G being so negative in the forward reaction, this would require that the delta G in the reverse reaction is absurdly positive, and we don't want that. That's energetically unfavorable. So we use just a whole different set of enzymes to do this. And so I would like to talk about the regulation of the key enzymes that I've highlighted in yellow if you want to rewind the video. So the first one being pyruvate carboxylase. So here, acetyl-CoA serves as an activator of pyruvate carboxylase. And I would like to highlight that in the pyruvate dehydrogenase complex, acetyl-CoA serves as an inhibitor. And this makes perfect sense, right? If we have a buildup of acetyl-CoA, it emphasizes that we're in an energetically satisfied state. We don't need more acetyl-CoA, but this buildup can also be used to indicate that we can start converting our focus from production of ATP to storage, such as the production of glucose that can ultimately form glycogen. So that is why acetyl-CoA in high concentrations activates pyruvate carboxylase, but inhibits pyruvate dehydrogenase complex. The next enzyme, the rate-limiting enzyme of gluconeogenesis, is fructose 1,6-bisphosphatase. So here, it's basically the reverse of PFK1. High ATP indicates that we should activate this enzyme, and this makes perfect sense. If we're high in ATP, we don't need to be focusing on producing more. We should instead be focusing on storing more molecules so that we have energy stores for all kinds of processes. And so high ATP indicates that we should activate fructose 1,6-bisphosphatase, which would also indicate for the inhibition of PFK1. Likewise, a buildup of AMP and a buildup of fructose 2,6-bisphosphate indicates to inhibit fructose 1,6-bisphosphatase. And this makes perfect sense. If we're low on energy, why are we going to spend that energy on making glucose? Instead, we should be spending it on glycolysis through PFK1 to start trending into the citric acid cycle and all of these aerobic respiration and aerobic production of ATP. This I wrote here to emphasize because you're going to see this a billion times in here. Every inhibitor in carbohydrate metabolism or every activator is a reflective representation of the energy state of that cell. I'm going crazy with this laser pointer right now because everything is about the energy state of the cell for carbohydrate metabolism. The next form of regulation is in glucose 6-phosphatase, which functions to remove the phosphate from the 6th carbon of glucose. This, I would like to note, is only found in the endoplasmic reticulum lumen of the liver, and this allows the release into the blood. And so I'd like to raise the point here that gluconeogenesis in the muscle and the kidneys and all this stuff is typically to produce glucose that will function as energy for that cell and that cell only. The liver, however, performs gluconeogenesis to release glucose into the blood, such that the blood and the glucose can travel to cells that need it, like the brain, which loves to use glucose. Here, that means that the liver is not actually getting energy from gluconeogenesis. Instead, it's using energy from beta oxidation of fatty acids as its energy source. Anyway, that summarizes gluconeogenesis and glycolysis. I hope you can see the similarities and differences. The regulation similarities and differences are essentially reciprocals. I would like to go over some less important pathways, heavy emphasis on the quotes here, because they're not really less important. They're just more like feeder pathways that fit into the two big ones I've just went over. And these are going to be glycogenesis and glycogenolysis, the pentose phosphate pathway, and then fructose and galactose metabolism. I'm not going to spend too much time on these, but it is important to know. So anyway, here is glycogenesis and glycogenolysis. I would like to start that by introducing that the feeding point from glycolysis or gluconeogenesis is glucose 6-phosphate, the second step of glycolysis, or the second to last step of gluconeogenesis. Here, this is like a cycle between going from glucose to glycogen to glucose. But know that glucose 6-phosphate is the crossroads for this process. Here, glucose 6-phosphate will turn into glucose 1-phosphate via the enzyme phosphoglucomutase. All it's doing is moving the phosphate group. Here's where it gets a little tricky. Not really, but it's just a little nuanced. Glucose 1-phosphate turns into UDP glucose. UDP meaning uridine diphosphate, just a nucleotide. It uses UTP and produces PPI, which is pyrophosphate. The result is that the enzyme UDP glucose phosphorylase is what catalyzes this reaction. Know that UDP glucose is the precursor for creating glycogen with the two enzymes that we're all aware of, glycogen synthase and branching enzyme. Now, glycogen synthase will create the alpha-1,4 glycosidic linkages in glycogen, whereas branching enzyme will create the alpha-1,6 glycosidic linkages in glycogen that give it its notorious branching. The enzyme glycogen synthase is the rate-limiting enzyme of this process, and it is activated by a buildup of glucose 6-phosphate and insulin. And this makes perfect sense because insulin is released when blood glucose is high. When blood glucose is high, we can expect that more glucose is entering the cell, which would result in a buildup of glucose 6-phosphate, which would tell hexokinase, hey, slow down, we don't need any more. Let's use this glucose 6-phosphate. And one of the ways we would use this glucose 6-phosphate is for producing glycogen. So that should be a light bulb in your head explaining how this all makes sense. And furthermore, it is inhibited by epinephrine and the hormone glucagon, which acts as an antagonist to insulin. Epinephrine kind of, triggers the fight or flight, which is like the breakdown of glycogen for whatever we're doing. So it would obviously prevent building of glycogen. And then glucagon basically says we need to raise the blood glucose levels, so we're going to break glycogen. So anyway, from the point of glycogen, we would then perform glycogenolysis to break it into usable glucose. This is done by the enzyme glycogen phosphorylase and debranching enzyme. Basically the opposites of these two. What activates glycogen phosphorylase, which is the rate-limiting enzyme of glycogenolysis, is basically reversals of glycogenesis. A buildup of AMP indicates that energy is low, and we need to start breaking down glycogen into mobile glucose, so this makes perfect sense. In the same way that glucagon inhibited glycogen synthase, glucagon will activate glycogen phosphorylase. In the same way that epinephrine inhibited glycogen synthase, epinephrine will activate glycogen phosphorylase. Here, ATP will inhibit glycogen phosphorylase, and this makes perfect sense. If we have tons of ATP, we're already energetically satisfied. Why would we break our storage to produce more glucose that we're not going to use? This will act as an inhibitor. The breakdown of glycogen by these enzymes results in glucose 1-phosphate, which, as you can see, is the same glucose 1-phosphate here, which uses the same enzyme to catalyze the forward and reverse reactions. This glucose 6-phosphate can feed into or out of glycolysis and gluconeogenesis depending on the energetic demands of the cell. Next, I would briefly like to talk about the pentose phosphate pathway, which is really, I've never seen even a practice question about this, but it still is good to know. The pentose phosphate pathway also feeds from glucose 6-phosphate, and the end goal here is ribose 5-phosphate and NADPH. Ribose 5-phosphate is a precursor for nucleotide biosynthesis, which is obviously very important for the integrity and maintenance of our genome. And then NADPH serves as one of the body's most natural antioxidants to reactive oxygen species. The nitty-gritties of this process is a little bit annoying, so I would like to just really emphasize that the enzyme that produces this NADPH is glucose 6-phosphate dehydrogenase. If you would like to pause and follow this process to ribose 5-phosphate, you can. The last thing I would like to emphasize is that there is a pool of sugars of varying carbons, like 4, 5, and 7, that are all in equilibrium, and they are all interchangeable between ribose 5-phosphate and these glycolysis and gluconeogenesis intermediates. They use the enzymes trans-ketolase and trans-aldolase, and that's really all that needs to be said about the pentose phosphate pathway. Heavy emphasis on nucleotide biosynthesis and antioxidant properties of NADPH. Next, we have fructose and galactose metabolism. So fructose comes from sucrose as well as fruit and honey, but I've wrote sucrose and fructose. What we would do once we have fructose is we would phosphorylate fructose in the same way we phosphorylate glucose. We'd create fructose 1-phosphate through the enzyme fructokinase. From here, fructose 1-phosphate can be cleaved into 2 3-carbon molecules in the same way that it is in glycolysis. It uses the enzyme aldolase B to create DAP, and this is the same DAP we've seen, and also glyceraldehyde. Glyceraldehyde is not the same as glyceraldehyde 3-phosphate, which it will ultimately become. Both DAP and G3P can enter glycolysis, gluconeogenesis, or glycogenesis. Really depends on the energetic demands of the cell, but this is fructose metabolism, not very complicated. Next, we have galactose metabolism. We get galactose from lactose, which is a sugar, a disaccharide of galactose and glucose, typically found in dairy and milk and all that stuff. Anyway, once we have galactose, we want to trap it in the cell in the same way we do all of our monosaccharides. We phosphorylate it with galactokinase. Galactose 1-phosphate will turn into glucose 1-phosphate through a series of reactions that use 2 enzymes, galactose 1-phosphate uridyl transferase and UDP galactose epimerase. Recall that galactose and glucose are C4 epimers, which means they vary at only 1 carbon, and so these enzymes result in a transformation into glucose 1-phosphate. From here, glucose 1-phosphate has a lot of options. It can enter glycogenesis because if we recall from glycogen metabolism or glycogenesis and glycogenolysis, glucose 1-phosphate turns into UDP glucose, which turns into glycogen. Or glucose 1-phosphate can turn back into glucose 6-phosphate and enter these pathways. So when we're here with galactose and we have glucose 1-phosphate, we can either enter glycogenesis like I said, or we can keep using the phosphoglucose mutase to turn into glucose 6-phosphate. And from here, we can enter glycolysis or gluconeogenesis. Again, all dependent on cellular demands, but these are options. Finally, I would like to say we are almost halfway done with this. Everything before is anaerobic, the lame stuff. And I say lame because if O2 is available, oxygen, we have aerobic processes, and these produce a ton more ATP. And so the 3 processes I'll be talking about are the citric acid cycle, the electron transport chain, and oxidative phosphorylation. The citric acid cycle serves for the purpose of creating more electron carriers. The electron transport chain serves for the purpose of creating a proton gradient, and oxidative phosphorylation serves the purpose of creating a ton of ATP. So this is really the fancy stuff, and we're going to cover all of this. So the first thing we'll cover is the citric acid cycle. This is a diagram I made when I originally was studying for the MCAT, and so I'm going to walk you through it. This covers essentially everything except for regulation, which is going to be the next page. Before, I would like to emphasize, though, that the citric acid cycle is technically oxygen independent. There's not a single step in this cycle that requires oxygen, but I put not really, because I also think you guys, if you think for a second, can really understand this. The citric acid cycle creates these intermediates like NADH and FADH2 that feed into the electron transport chain. The electron transport chain is oxygen dependent. So if oxygen is low, the electron transport chain will slow down, which would lead to the accumulation of these electron carriers. And you'll learn soon that the buildup of these electron carriers inhibits the electron transport chain. So we kind of have a domino effect where if the electron transport chain slows down, the citric acid cycle will slow down as well. But anyway, the citric acid cycle serves to do one very important thing, and it's the production of high potential electron carriers in the form of NADH 3 times, which I'm circling here, and FADH2 just once, and we do get an actual production of ATP, a physical production of ATP, and this is the only time this ever happens in the citric acid cycle. The citric acid cycle takes place in the mitochondrial matrix. Red blood cells lack mitochondria, which means they don't do this. And so this only takes place under aerobic conditions. The cycle, heavy emphasis on cycle because it's not a pathway like glycolysis. This is happening over and over and over again. This begins with pyruvate turning into acetyl-CoA, which is a 2-carbon molecule, and oxaloacetate, which is a 4-carbon molecule. Acetyl-CoA, like I said, can come from many things like ketogenic amino acids like leucine and lysine, ketones, ethanol, and fatty acids. This could all be went over with amino acid metabolism and lipid metabolism because that involves like fatty acid shuttles and the carnitine shuttle and all of that. And quite frankly, that's not too important. Just know that we have acetyl-CoA somehow. Acetyl-CoA, oxaloacetate, 2-carbon, 4-carbon. This will use the enzyme citrate synthase, which is a condensation reaction requiring water, and it will create citrate, which is a 6-carbon molecule. Citrate will then turn into isocitrate through the enzyme cis-aconitase. This requires iron as a cofactor, and all this does is it rearranges a hydroxyl group, an OH group, on citrate and isocitrate. From here, isocitrate will become alpha-ketoglutarate through the enzyme isocitrate dehydrogenase. I emphasized here, this is the rate-limiting enzyme, and we'll talk about that real soon. This reaction results in the production of 2 molecules. One is a CO2 molecule, so decarboxylation has occurred, meaning we've gone from 6 carbons to 5 carbons, as well as an oxidation reduction that resulted in a reduced NADH being formed. And this is the first of 3 NADH that will be formed in the citric acid cycle. From here, alpha-ketoglutarate will turn into succinyl-CoA through the enzyme alpha-ketoglutarate dehydrogenase complex. This complex requires similar or the same cofactors as the pyruvate dehydrogenase complex. Also note that we are having the second and final decarboxylation occur, and we're having the second of 3 productions of NADH. So we've gone from a 5-carbon down to a 4-carbon succinyl-CoA. From here, we will go from succinyl-CoA to succinate, and we will use the enzyme succinyl-CoA synthetase. Now, I would like to really point out here that this is the only time we're producing a molecule of ATP directly, and the reason we can do that is because of coenzyme A. Coenzyme A has a thioester, which is very high in energy, such that when it's broken off of succinyl-CoA to form succinate, that energy is coupled to the phosphorylation of GDP into GTP. That GTP can then be used to transfer its phosphate group to an ADP, which will turn it into ATP. The enzyme that does this is nucleoside diphosphate kinase. Just know that the breaking of the coenzyme A results in the release of high energy from the thioester bond, which couples to the phosphorylation of GDP. Succinate will then turn into fumarate through the enzyme succinate dehydrogenase. This is known as complex 2 in the electron transport chain, which we'll talk about real soon. Succinate will result in FAD being reduced into FADH2. And the reason we use FAD instead of NAD+ is because the reducing power of succinate dehydrogenase is not strong enough to reduce NAD+. So instead it uses FAD, which is a flavoprotein. FADH2 still gets a job done and it produces ATP, albeit less than NADH. From here, fumarate will turn into malate through the enzyme fumarase. This is a hydrolysis requiring a molecule of water. And so from here, malate will turn into oxaloacetate again through the enzyme malate dehydrogenase. This is the third and final production of NADH. And so I've written a tally here. We've made directly 1 ATP through this step here. We have made 4 NADHs, and I say 4 because one of them came from the production of acetyl-CoA. And we have made 1 FADH through succinate dehydrogenase. NADH produces approximately 2.5 ATP per molecule. FADH2 produces approximately 1.5 ATP per molecule, which means all of this for 1 pyruvate molecule is around 12.5 ATP per pyruvate. But recall that glucose creates 2 pyruvate molecules, which means 1 glucose molecule can result in 2 cycles of the citric acid cycle. And so this means 25 ATP because you multiply the 12.5 by 2. This is just the net reaction I wrote. I don't think you'll ever need this, but it is good if you're a chemistry nerd and you want to keep track of this. Here is the regulation of the 3 enzymes in the citric acid cycle. So number 1 is citrate synthase. This is inhibited by high levels of citrate and succinyl-CoA, so a form of feedback inhibition, as well as when the cell is in a high energy state, which is indicated by high ATP and high NADH. This all results in inhibition. The rate-limiting step of the citric acid cycle is isocitrate dehydrogenase, which is inhibited by high levels of ATP and high levels of NADH. Again, energetic state of the cell dictates this, and it's activated by high levels of ADP and high levels of NAD+, which means it's activated by a low energy state. So again, big trend. I've highlighted it a million times now. Energetic state of the cell dictates carbohydrate metabolism. The third and final enzyme that's regulated in the citric acid cycle is the alpha-ketoglutarate dehydrogenase complex. Same thing here. We have some feedback inhibition here. The product, succinyl-CoA, will inhibit the enzyme. And the same thing, high energy state of ATP and NADH indicate that alpha-ketoglutarate dehydrogenase has to shut down. Again, trend. ATP and ADP ratio and NADH and NAD+ ratio dictate the metabolic process of a cell. Here we have the electron transport chain covering all four complexes, from complex 1, complex 2, complex 3, and complex 4. These are a little confusing, so I will explain them, but know these things in green are actually the most important. Complex 1 uses NADH, and what it does is it oxidizes NADH from the citric acid cycle into NAD+. The electrons from NADH are going to go to a coenzyme, FMN, which is another flavoprotein, and this will form FMNH2, so it reduced FMN. This reduced FMNH2 is going to move its electrons to a iron-sulfur cluster, which is currently oxidized, but this moving of electrons will turn it into a reduced iron-sulfur cluster. This reduced iron-sulfur cluster will finally move its electrons to the coenzyme Q molecule, also known as ubiquinone, and this moving of electrons reduces it to coenzyme QH2, also known as ubiquinol. This process results in the pumping of 4 protons from the mitochondrial matrix into the intermembrane space. Complex 2, a very similar process. Recall that it's succinate dehydrogenase. It's going to take succinate and oxidize it, creating fumarate. And this oxidation is coupled with a reduction of FAD into FADH2. FADH2 will reduce the same iron-sulfur clusters we've seen into reduced forms of the iron-sulfur clusters. And this reduced iron-sulfur cluster will once again transfer its electrons to coenzyme Q to create coenzyme QH2. No protons are pumped in complex 2. Complex 3 is the one that gets a little confusing because it's where the Q cycle occurs. The Q cycle is how we take this coenzyme QH2 and actually move the electrons to cytochrome C, which is going to, kind of fast forward here, is going to end up being what reduces the oxygen. Basically, though, what the Q cycle is we use one coenzyme QH2 and a second one. And what happens is the 2 electrons in coenzyme QH2 are split up. One of them goes to cytochrome C to reduce it, and the other forms a semiquinone intermediate, which is just 1 electron on a coenzyme QH, really, not H2 then. And so then a second coenzyme QH2 is going to do the same electron splitting, and it will create a new coenzyme QH2 because it will fulfill the additional electron requirement of the semiquinone intermediate, but it will also reduce a second cytochrome C. So in a single Q cycle, we are producing a brand new coenzyme QH2 and 2 cytochrome Cs in the reduced state. However, we need 4 reduced cytochrome Cs to ultimately create water. So we're going to need 2 Q cycles to occur. But do note that this results in the net pumping of 4 protons into the intermembrane space. Complex 4 is the complex that allows the final electron acceptor, diatomic oxygen, to donate 4 electrons to create 2 water molecules while simultaneously recycling cytochrome C back to its oxidized state. This complex results in the pumping of only 2 protons. Once again, though, notice that we are pumping tons of protons into the intermembrane space to create a gradient. And you may be asking what that gradient does. And so with that, we're going to talk about it right now. Oxidative phosphorylation and chemiosmotic coupling are very important to understand. I've simplified the electron transport chain here to show that we've pumped protons into the intermembrane space. This is the outer membrane, this is the inner membrane, and this is the mitochondrial matrix. ATP synthase is here, downstream from this. And so ATP synthase has two domains that you should be aware of. One is the F0 domain, and the other is the F1 domain. The F0 domain is responsible for capturing the energy of the proton gradient we've established, and the F1 domain is the actual machinery that phosphorylates ADP into ATP. And so how are we taking this gradient and using it? Well, the pumping of protons creates an electrochemical gradient. And what this means is, we have tons of protons in the space, which means the pH has gone down. We have a charge difference. All of this stuff leads to wanting to move down the gradient. And we know that moving down gradients creates energy. We've seen this in secondary active transport. This gradient, once it moves through the F0 domain, will provide tons of energy to phosphorylate ADP. And so I wrote here, the delta G of chemiosmotic coupling is around negative 220 kilojoules per mole, which is absurdly energetically favorable. And so this is enough energy to fuel a ton of ATP production. And so we're going to talk about the tallying of that ATP. But first and foremost, I would like to say that oxygen and ADP dictate all of our metabolic processes. And think about this. I did allude that oxygen indicates if the electron transport chain will be performing efficiently or not. And if the electron transport chain isn't performing efficiently, we get an accumulation of electron carriers, which leads to inhibition of the citric acid cycle. So oxygen is the first and foremost highest dictator of aerobic respiration. And that makes perfect sense because aerobic means requiring oxygen. ADP, though, is the next most important, because if we are in a state of supplemented oxygen and we're happy, we're satisfied with our oxygen, our ADP is the next indicator. We have oxygen, but how much energy do we have? If we have high ADP, we're low in energy, and we're going to really want to perform oxidative phosphorylation. If we have oxygen and we're low in ADP, we're golden because we're breathing well and we have tons of energy. So these two molecules dictate everything. I would like to do a final tally of ATP produced as a result of anaerobic and aerobic metabolism. Recall that in glycolysis, I told you to keep these two numbers in the back of your head. We produce 2 net ATP and 2 NADH, which each produce 2.5 ATP per molecule. So that's 2 plus 5. We're totaling 7 in glycolysis or anaerobic metabolism. In the citric acid cycle, we are getting 1 NADH from the production of acetyl-CoA by pyruvate, which results in 2.5 ATP. We had 3 NADH and 1 FADH2 made in the actual citric acid cycle, totaling 7.5 and 1.5 respectively. And then we directly made 1 ATP from our conversion of succinyl-CoA to succinate. And so this means we have per pyruvate molecule 12.5 ATP. But 2 pyruvate molecules come from 1 glucose, which means 1 glucose results in 25 ATP in the citric acid cycle. When you add these two numbers together, you get around 30 to 32 ATP per glucose. Without going into too much detail, I would like to also notify you that the reason we say 30 to 32 and not just definitively 32 is because this is really dependent on how effectively a cell is performing glycolysis. Some of them, there's 2 shuttles that allow cytoplasmic NADH to contribute to the electron transport chain, the glycerol 3-phosphate shuttle and the malate aspartate shuttle. Both of these are effective, but the malate aspartate shuttle is a lot more effective in providing this NADH instead of FADH2. And so depending on what shuttle and how effective the cell is at glycolysis will be if we get 30, 31, or 32, or maybe even more. But anyway, we say 30 to 32 to be safe. And so that is all. That really should be all you need to know about carbohydrate metabolism. This is a very dense video. I may have talked fast, and some of the things I said were not on these notes, but you can screenshot them, write on top of what you've already been note-taking. But in some way, we have covered everything that the big third-party companies would cover in a little bit under an hour. So hopefully we'll have more of these coming out. And so this is the first one. I found it fitting that we would cover one of the most notoriously high-yield topics first, just to kind of give you a gist of how my teaching style is and how we cover this material. So stay tuned for more.